Dr. Pellman, you're muted. Sorry about that.
Okay. I'm David Pellman, and it's a real pleasure today to introduce Scott Keeney. Scott Keeney from Memorial Sloan Kettering. Scott is going to be delivering the Louise and Herbert Shivek lecture in our Seminars in Oncology series today. This is actually going to be our last seminar for this year before the summer break. For nearly 40 years, the Shiveks have been dedicated donors and volunteers of Dana-Farber. In 2006, they made a substantial gift to establish the Louise and Herbert Shivek Lecture to fund an annual seminar in basic science. The Shiveks' belief in educating the next generation of oncologists and they fully support all cancer research efforts. We're tremendously grateful to Mr. and Mrs. Shivek for all they have done for Dana-Farber over the years, and we're proud to have an annual seminar which bears their name. And I want to extend particular thanks to members of the family who, as I understand it, are listening in today. I believe that we have the perfect speaker for the vision of what the Shivek lecture should be, and that's Scott Keeney. Word of background, Scott was a Phi Beta Kappa graduate of Virginia Polytech, who then did his PhD at UC Berkeley, and then moved to Boston, where he did a postdoctoral fellowship at Harvard University with Nancy Kleckner. Scott has been, for his entire faculty career, been at Memorial Sloan Kettering, where he currently holds an endowed chair. Scott's received many honors, too many for me to list, but in 2008, he became an investigator at the Howard Hughes Medical Institute, and he's an elected fellow of the American Academy of Arts and Science, and was recently also elected to the National Academy of Sciences. Scott has made tremendous contributions to our understanding of meiotic recombination. In meiosis, cells are essentially playing with knives and need to make controlled DNA double-stranded breaks that are distributed in a certain pattern and at a certain time. And Scott's group has used really innovative combinations of genome scale approaches along with mechanistic biochemical approaches to provide leading insights into how this works, which we're going to hear today. One final word, which is on process, which is that Scott will give a seminar, and at the end, I'm going to ask everybody to please post their questions in the question and answer. We're not going to be able to raise hands, and in whatever time that remains, I'll read those questions, and we'll let Scott answer as many as possible, and the others he'll get and perhaps may be able to answer by email. So without further ado, thanks very much, Scott. We're looking forward to the seminar.
Thanks, David. I really appreciate the very generous introduction. And I also very much appreciate the invitation. And especially want to thank the Shiveks for their generosity in supporting this lecture series. And I'm very honored to have a chance to present our research as part of this series. So, sorry, I want to start a timer so I don't run over. I want to tell a couple of stories today that relate to this issue of how cells create the machinery to make double-strand breaks in the DNA. And most of us, of course, think often about double-strand breaks in other contexts than in the context I will be talking about. And in those other contexts, for example, in somatic cells, double-strand breaks are usually a dangerous thing. But during meiosis, as I will explain, double-strand break formation is actually essential for normal development. And it's a rather fascinating aspect of how cells kind of play with fire with their own genomes. So as you all know, most of us probably don't think about this that much from except when we were undergrads. But as you all know, we all went through a pair of very severe genetic bottlenecks when cells in our parents reduced their genome complement from the normal diploid state in most of the cells in our body to the haploid state to generate reproductive cells, either an egg or a sperm, so that when the egg and the sperm could then fuse to generate the zygote that would then develop into the new person, the genome complement was restored to the diploid status. And I've been very interested throughout my career in this really fascinating reduction in the genome complement from diploid to haploid. So to orient what I want to talk about today, I first kind of compare and contrast meiosis to mitosis, which again, probably more people are more intimately familiar with. So of course, the goal of mitosis is to generate daughter cells that have the same genome complement as the starting cell. So what's cartooned here is a very simple diploid that has a pair of homologous chromosomes. So you can imagine it inherited the red chromosome from its mom and the blue chromosome from its dad. And each dividing cell in the body is going to generate two daughter cells that have that same genome complement. So this is accomplished by doing one round of DNA replication to generate a pair of sister chromatids for each chromosome. And then those sister chromatids are pulled apart from each other. And then after nuclear division and cytokinesis, you end up with two daughter cells. Goal of meiosis is different. As I mentioned, the goal of meiosis is to reduce the genome complement. So you're going to generate now haploid products for packaging into reproductive cells. And the way cells do this is to do one round of DNA replication, but now two successive rounds of chromosome segregation. The second round of meiotic chromosome segregation is a lot like a mitotic one, so that's why I have it cartooned on top of the mitotic division, because you can see what's being pulled apart are the sister centromeres. It's this first division that's unique to meiosis because what's pulled apart here is pairs of homologous chromosomes. So at its heart, this is a DNA sorting problem, right? The cell has to be able to figure out which chromosomes are homologous to each other so that it knows which chromosomes to segregate from one another. And in the simple diploid, that's not much of a task because there's only two chromosomes, but most real organisms have a much more complicated karyotype. So budding yeast, for example, has 16 pairs of chromosomes. We have 23 pairs of chromosomes. So there's sort of a spaghetti bowl of DNA molecules in the nucleus that all have to be sorted into pairs so that those pairs can then be separated from each other. So the name of the game in meiosis is this sorting where the chromosomes actually line up with each other along their lengths and exchange a little bit of genetic information through the form of this crossover recombination product. And it's this pairing and recombination that allows the homologous chromosomes to recognize one another as homologs and allows the cell to segregate them accurately from one another. So as it's cartooned here, you can see that meiotic chromosome segregation is quite a bit more complicated than mitotic chromosome segregation. So this being Pride Month, we can sort of vision that this is mitosis and its more flamboyant friend, meiosis. And your mileage may vary, but I tend to think of meiosis as the more exciting version of the cell division. Okay, so for our purposes, we're going to focus on this early part of the process after DNA replication, what the cells need to do in order to achieve this pairing and recombination. So as it turns out, most species accomplish this by deliberately damaging their DNA. So these little green stars are supposed to represent a type of DNA damage that has to be repaired by searching for a homologous DNA duplex that can serve as a template for the repair reaction. That search for homology is what actually allows the homologous chromosomes to line up with one another. We know a lot about how this DNA damage works and is repaired from studies in budding yeast. So now what you're looking at is a cartoon of the DNA events during meiotic recombination. So each line here is now one strand of DNA duplex. So this is one DNA duplex. You have to imagine there's another red sister chromatid that I'm not showing you in this cartoon. And the initiating event, these little green stars, that's a double strand break that's made by SPO11. SPO11 is a topoisomerase relative, as I will discuss in quite a bit more detail in a little bit. And like other topoisomerase-like proteins, it cuts DNA through making a covalent attachment to the DNA strand that it has attacked. In order to then repair these double-strand breaks, the cell has to process the ends, and it does that by nicking the DNA strands that SPO11 is attached to. That releases SPO11 with these little oligonucleotides attached, and frees up the end for further processing by exonucleases that yield at the end 3' single-stranded tails, which can be coded by strand exchange proteins, which will allow this single-strand DNA to search for a homologous DNA and invade it. After then a series of fairly complicated and highly regulated steps, the double-strand break can be repaired in either of two outcomes. One outcome that we call non-crossover involves just copying a little bit of information from the intact donor chromosome, so you get this patch-like repair. But alternatively, the cell can build a more complicated branch DNA structure called a double holiday junction, which can then be resolved to yield that crossover product that holds the chromosomes together temporarily until the first division. So what I want to focus on today is this very earliest step, the formation of double-strand breaks. And SPO11 does not work by itself in Saccharomyces, where this has been studied probably in most detail. We know that there are at least 9 other proteins in addition to SPO11, so 10 in total, that are absolutely essential for double-strand break formation. And what's cartooned here is an interaction map based on yeast-2 hybrid and other studies over the past 20 years or so that's led to an understanding of how these proteins relate to one another. So here's SPO11 itself, and based on these interaction maps, we've sort of defined that there's this set of proteins that interacts very closely with SPO11 that we call the core complex, and this is what we think is the enzymatic heart of the double-strand break machinery. Then there's a set of proteins that we refer to as RMM for REC114, MY4, and MER2. And these are a looser association of proteins, or at least seem to be a looser association of proteins that was distinct from the core complex. There's also the RAD50-MRE11-XRS2 complex that's critical in yeast, but its role is not conserved for double-strand break formation. In some species, it's required for break formation, but in others, it's not. And I'm not going to dwell on that one for the purposes of the talk today. So what I'm going to talk about today is these two complexes. So first, let's start with SPO11, since that's sort of the heart of the machinery. So as I mentioned, SPO11 is related to topoisomerases, and in particular, it's related to this archibacterial topo called topoisomerase 6. So first, let me tell you a little bit more about topo 6. Topo 6 is made up of 2 subunits called the A subunit and the B subunit. The A subunit has 2 domains, a metal binding domain called a toprim domain, and a winged helix domain that's sometimes referred to as a 5Y cap domain. This domain contains the tyrosine, the Y is reference to that tyrosine, that is the catalytic residue that actually cleaves the DNA and becomes covalently attached to the DNA end. The B subunit has a domain called a GHKL domain, which is an ATP binding, ATPase, and ATP-dependent dimerization domain. Then there's an alpha helical domain that's called the transducer that is thought to transduce information from the ATPase module to the DNA cleaving module of the A subunit. So the way the topoisomerase is thought to work is that one DNA duplex is bound to 2 A subunits, and with the B subunits open, then a second DNA duplex can be captured, and upon ATP binding, dimerization of the GHKL domains can trap that second DNA duplex along with the first DNA duplex inside this ring-shaped structure. ATP hydrolysis is thought to then trigger DNA cleavage and opening of a gate in the DNA that then allows that second DNA duplex to pass through. And this, of course, is the topology change reaction that the enzyme is trying to catalyze. But because of that covalent complex of the protein with the DNA ends can be religated to one another isoenergetically with the cleavage reaction, and that allows restoration of the original DNA duplex without a frank double-strand break. Then ADP release can open up the GHKL domains from each other and recycle the enzyme to go through another cycle. Okay, so again, here's topo6 in its domain architecture. It's found in archaea. It's also found in some green plants where it plays roles in endoreduplication in some tissues. Most eukaryotes, though, have lost a bona fide topoisomerase version of topo6 and instead have retained this protein family only in the form of this meiosis-specific protein SPO11. So cartooned here are SPO11s from various species, from mouse, Cerevisiae, where it was originally discovered, Pombi, flies have a different name, and Arabidopsis actually has two versions of SPO11 along with its topo6 version. We don't really know exactly why there are two SPO11s in Arabidopsis, but it's thought perhaps they make a heterodimer. So the domain structure of SPO11 looks very much like the domain structure of topo6A. For many years, it was a bit of a puzzle as to whether there was an equivalent of the B subunit. But a really beautiful pair of papers from Mathilde Grelon's lab and Bernard Demassi's lab identified for the first time the equivalence of the B subunits in mice and in plants. And I'll probably mostly refer to this as top6B-like, which is the mouse name. So the mouse and plant proteins had clear matches to what looked like a transducer domain and a GHKL domain, but I've cartooned the GHKL domain as kind of a mangled version because this GHKL domain in these proteins is lacking some residues that are critical for using ATP and ATP hydrolysis to govern dimerization. So it's not actually clear that they can bind ATP or hydrolyze it. These papers also identified equivalents of these proteins in other species, and but interestingly, those versions were equivalent apparently only to the transducer domain. There was no equivalent to the GHKL domain in these proteins. And one of these proteins, the one in Cerevisiae, is called REC102, which actually was on that earlier interaction map that I told you about. It was as one of the core partners of SPO11. So that was really interesting to us. And we're also kind of intrigued by the absence of this GHKL domain. Okay, so here's the core complex of yeast SPO11. With SPO11, there's that REC102 protein I just mentioned. But then there's two other proteins that we didn't really know quite how they fit in here, REC104 and SCI8. So for many years, this is the resolution that we had for information about the SPO11 core complex, just kind of circles and arrows to kind of connect them to each other. We tried really hard to purify these proteins and really met with no encouraging successes at all for really 2 decades. It was only a few years ago then that this problem was cracked by this really talented, now former postdoc from my lab, Corentin, who has his own lab in Belgium now. And he, for the first time, was able to purify the SPO11 core complexes from, this is the yeast proteins expressed in baculovirus vectors in insect cells. And so here's two different preparations. One of them has an maltose binding protein tag on REC102. The other has no MBP tag. And you can see here's the SPO11 protein, which is tagged with His6 and flag epitope. Here's the SCI8 protein. Here's REC102, either without the tag or with the tag. And here's REC104. And these are actually present in equimolar ratio. The REC104 just stains with Kamasi less well. We have other data that shows that these are in an equimolar ratio. So one of the first surprises was that actually this complex is monomeric in solution, by which I mean it had a 1 to 1 to 1 to 1 stoichiometry, not 2 to 2 to 2 to 2. This was a surprise because the archaebacterial enzyme is constitutively a dimer through stable interactions of the A subunit, which is the equivalent of SPO11. But these SPO11 complexes do not seem to stably make the expected heterooctamer of 2 subunits of each. Instead, they make this sort of what I'll call a monomeric species. And here's some of the evidence for that. So this is Sekmal's experiments showing the elution of the untagged complex in blue and the tag complex in red. And the molecular weights of these complexes and also the difference in molecular weight between the tagged and the untagged are exactly what you would expect for monomeric complexes rather than dimeric. We can also look at these by negative stain EM, and no matter which subunit we put the maltose binding protein tag on, we only see one extra bit of density on the tagged complex relative to the untagged complex. And this density is appropriate dimensions to be 1 mbp. So it is pretty clear that, at least under the conditions we've looked at, this complex is not dimeric. Okay, so here's again those negative stain EM images, and this is kind of our interpretation of what we're looking at here, where we think that this part of the density is SPO11 and SCI8, and this part of the density is REC 102 and REC 104. And I'll now kind of explain why we think that's the way it is, and leave you with a structural model for how this might be assembled. Okay, so for the next few slides, I'm going to be presenting SPO11 and its partners in this structural model, and I'm going to show it as if it's a dimer, even though I just told you that it's not dimeric in solution, because when it cuts DNA, it does have to be a dimer, because you need 2 SPO11s, 1 to cut each strand. So it must work as a dimer and come together at some point, but at least in solution, the recombinant proteins don't behave that way. So here's what topo 6 would look like, and we're just modeling the SPO11 REC 102 structure based on that. This is sort of the cartoon form. And from a variety of other studies, we already had the idea that SCI8 probably docked onto the complex like this. SCI8 is a WD40 repeat protein. It makes this kind of donut, shaped structure. And we were pretty sure exactly where it docked onto the structure. So here's a space filling model, 2 different orientations of what we think this looks like. I want to emphasize this is a homology-based model. So we've taken the crystal structure of the archaeal enzyme and we've built a homology model based on that. So this is partially fantasy. We certainly do not have this high resolution image of what SPO11 looks like, but this is to provide a framework for talking about the structural studies that I'll talk about. So the DNA would be expected to kind of bind in this groove here, and the transducer domains poke up out of the plane of the slide in this part of the image, and SCI8 is docked here, as I said. Okay, so here's a ribbon representation of that same structure. Okay, so the first way we sort of tried to probe into this structure was to evaluate the connectivity between the different polypeptides in the complex. And we did this in part by doing protein-protein cross-linking and mass spectrometry. So the idea here is quite simple. You have 2 proteins in a complex. You do covalent cross-linking between lysine residues on the 2 proteins, digest with trypsin, and use mass spec to identify which peptides have been linked to which other peptides. And doing that, you can get these nice interaction maps that report on what lysines in, say, REC 104 were in proximity to lysines on SPO11 and so forth. And from that, we can build up a picture of, kind of test predictions from that structural model that I just showed you. So one of the predictions was about where SCI8 docked. And very gratifyingly, on SCI8, there were 2 lysine residues that cross-linked very prominently to a lysine residue on SPO11. And this is the model from docking SCI8 onto SPO11 before we had the cross-linking data. And very nicely, you can see that the cross-links agree very well with what we would have predicted from this docking model. So that was nice to see, although the information that allowed us to dock it on is quite old. So this really didn't push us much further along, but it at least told us we were on the right track. So one of the new things that it told us was about REC 104. So this is kind of the orphan subunit here. We didn't know exactly where to place it in this complex, but we did know that it interacts very intimately with REC 102 and that it also interacts with SPO11 and SCI8. So where is it? The cross-linking data actually gave us a really interesting result. So here's in yellow is the model for REC 102. And there were two lysines on REC102 that were predicted to be in these positions that are very prominently cross-linked sites for REC104. And what's interesting about it is that this predicted location for these lysines corresponds to the position on topo 6b where the GHKL domain would sit. So these lysines are on the transducer domain, and in REC102, lysines in what are modeled to be equivalent positions are very prominent crosslinkers to REC104. So that seemed to indicate that in fact REC104 is sitting where the GHKL domain should be. But REC104 doesn't look anything like a GHKL domain that we've been able to tell, although some structure prediction programs can match it to a GHKL domain if you ask it to match it. So whether that's a good fit or not remains to be seen. But it did lead us to this idea that REC104 might be a highly diverged version of the GHKL domain. One possibility is it's actually evolved from a GHKL domain, but it's changed out of all recognition at the primary structure level. Another possibility is that it's actually a completely different fold that's replaced the GHKL domain. So to test this idea that that's where REC104 is in fact sitting, Corentin did alanine scanning mutagenesis across regions in REC102 predicted to then interact with REC104 based on this structural model. So the red residues are ones that he changed to alanine. And he did a yeast-2 hybrid experiment where he fused the mutant versions of REC102 to LEX-A, DNA binding domain, and then asked for interactions for REC104 fused to the GAL4 activating domain. And you can see that the wild type REC102 protein interacts very nicely with REC104, but a bunch of these different alanine scan mutations across this region in REC102 killed the interaction with REC104. Now this is an assay for recombination activity in vivo, where we're asking for recombination between two different mutant ARG4 alleles. And in wild type, you get lots of recombination between ARG4 when SPO11 is active. And so with wild type REC102, which you need for supporting recombination activity, you get this many ARG plus colonies out of the experiment. But in these mutants that are defective for interaction, you can see we kill the recombination activity in vivo. So this very nicely supports the idea that this interaction is critical for both double-strand break formation, but more specifically for the interaction between 102 and 104. So Corentin also probed another interaction surface on REC102 that we were interested in, which is the interaction surface with SPO11. So here's the predicted interaction surface between those two proteins, again, based on the archaeal structure. The red residues he mutated, and again, he got a similar result where he got lots of mutations that killed the interaction between REC102 and this time SPO11, and they also killed the function of the protein in vivo. So this seems to support between the cross-linking mass spec data and the structural modeling and the functional studies here, this supports the idea that SPO11, as we had anticipated, is likely to be conserve a lot of the structural aspects of topo 6. So the big question at this point is, can we reconstitute double-strand break formation in vitro? And unfortunately, the answer to that is not yet. So we haven't yet figured out how to do that. That's, could be really soon. It could be a long time from now, but we'll keep plugging away. In the meantime, what we have been able to look at is the interactions of the SPO11 complex with DNA, and that's actually proved to be quite interesting. So what you're looking at here are atomic force microscopy images of binding reactions of the SPO11 core complex bound to linearized plasmid DNA. And so if you just look at DNA only, you see these really nice traces for the DNA molecules on the mica surface. But if you incubate them with the SPO11 core complex, you can see these little particles associated with the DNA. And if we zoom in on a few of those particles, what Corentin noticed was 3 distinct DNA binding modes. One of those DNA binding modes involved SPO11 bound to the end of a DNA molecule. Another involved it binding in the middle of a DNA duplex. And interestingly, when it was bound in the middle of the DNA duplex, more often than not, the duplex was bent. So there seemed to be a preference for binding to bends in the DNA. But he also saw binding to junctions, either three-way junctions or four-way junctions, which, you know, this loop structure or two DNA duplex arms coming out of each side. And based on these studies and quantifying the data, he was able to deduce that the protein prefers to bind to ends best of all, and that if it's going to bind internally to a DNA duplex, it likes junctions of duplex better than it likes single duplex segments. And again, when it binds to single duplex segments, it seems to like to bind to bent DNA. So I want to focus a little more on this end binding activity, because that was quite surprising to us. It hadn't really been documented before for any other topoisomerases. And this is now a gel shift experiment where he's incubating a DNA substrate with increasing amounts of the protein. And there's two DNA substrates here, and I'll explain what those are. So each of those substrates is a hairpin. It's a DNA molecule that can fold back on itself, where the duplex part is 25 base pairs. And the non-hairpin end is either blunt or has a 2' 5' overhang, a TA sequence in this case. And with this titration, you can see very clearly, I think, that the protein really prefers binding to the overhang substrate. You can see at 0.4 nanomolar complex, you're already getting an appreciable amount of binding to this substrate and much less binding to this substrate. But as you increase the amount of the complex, you can see a second shifted band, and that second shift is occurring at about the same concentration in the two experiments. So because it's the same concentration with the two substrates, we infer that that second band is because of binding to the hairpin end, and that the first band is binding to the free end, and that it prefers the 2' 5' overhang. So the binding curves are quantified here, and again, you can see in blue, the TA overhang is preferred substrate. So he's measured apparent KDs for these interactions, and this is incredibly tight binding. It's sub-nanomolar affinity for substrate, these substrates, and it strongly prefers a 5' 2' 5' overhang, does not like a 3' 2' overhang anywhere near as much, or a blunt end. And competition experiments have shown that the affinity is too high for us to really distinguish by direct measurements on these isotherms, but by competition experiments, he can show that 2' 5' overhangs are definitely the preferred substrate of all of these structures. And that's interesting because 2' 5' overhang is in fact what SPO11 generates when it makes a double strand break. So I mentioned that SPO11 covalently attaches itself to DNA as part of the cleavage reaction. But what we're looking at here is non-covalent binding of the protein to the end. So in addition to being covalently attached to the strand, it's also non-covalently really clamped down on this duplex DNA end. And that was quite surprising to us because this had never really been documented before for other topoisomerases. We think it has some interesting implications for SPO11 function that I'll get back to at the end when I kind of give some more speculative models. Okay, so I told you about Topo 6 and its catalytic cycle. Topo 6 is intrinsically dimerized through the A subunit and then captures a DNA duplex that it binds, and it can capture a second DNA duplex. The thing I want to emphasize here is that the DNA duplex, whether it's the one that's going to be cut by SPO11 or it's the second one that's going to be passed through, sorry, not SPO11, Topo 6, or the second duplex is going to get passed through that DNA gate, those DNA molecules both enter through the ATP gate, the GHKL domain ATPase gate. But SPO11 does not seem to be intrinsically a dimer, and we're kind of entertaining the idea maybe SPO11 is inside out relative or upside down relative to Topo 6, and that maybe DNA molecules actually enter through the SPO11 gate rather than entering through the REC104 gate. And perhaps this gate is not controlled by ATP binding and hydrolysis because REC104 does not have any obvious ATPase motifs, but maybe this is controlled a different way. And the candidate for how that might be controlled is in this, this wiring diagram. It's, you can see that the REC102 and REC104 make very prominent interactions with members of this group of proteins, REC114, MY4, and MER2. So let me now tell you more about these guys, because we think they're potentially working this way to orient the SPO11 molecules. Okay, so this work was recently published a couple months ago, and so I'll go through part of the detail, though, to try to explain why we think these three proteins are particularly interesting. So before that, do that, I have to give you a little bit more background information about what's going on in the cells during break formation. So meiotic chromosomes form this very elaborate structure where there's a proteinaceous linear structure that we call the chromosome axis. And from that axis emanates out the chromatin of the chromosomes as loops. And we think that sister chromatids are co-oriented in these loops, but the exact relationship of the sister chromatids to each other isn't known. So the dashed line is supposed to be the sister of the solid line. So there are proteins that we can define as being structural components of the axis, and then there's the double-strand break proteins, which are these guys that are SPO11 core complex and SPO11 accessory proteins. And one of the things that's interesting is that we know that the DNA molecules that end up getting cut are the DNA segments that are out here on the loops, not the DNA segments that are embedded in the axis. But the double-strand break proteins themselves, these guys and the SPO11 core complex, they actually are enriched on the axis-associated segments. So how is it that the machinery parked here on the axis is cutting the DNA out here on the loops? The idea to explain that comes from models proposed by Nancy Kleckner and by Franz Klein with some input from lots of other labs, including ours, that leads to this model that we call the tethered loop axis complex model. And the idea is that double-strand break proteins are on the axis, but they capture DNA segments that are out on the loops, and they break those DNA segments then in spatial proximity to the axis. And I could talk later about why we think the cell wants to do that, to coordinate the break formation and repair with this higher order chromosome structure. But for now, just kind of take my word for it that this is sort of the paradigm model in the field for how this is organized in three-dimensional space. Okay, so Corentin also succeeded in purifying the REC114, MY4, and MER2 proteins, but these did not make a single stoichiometric complex. Instead, despite trying really hard, he could never get a complex with all three proteins, but he was able to get a complex that contained both REC114 and MY4. And he did cross-linking and mass spectrometry and was able to identify that the REC114 C terminus interacts predominantly with the MY4 N terminus. And he was able to show by a variety of methods that there's a stoichiometry of 2 REC114s to 1 MY4. So this very highly schematic cartoon image of what we think this heterotrimer might look like. Obviously, this is just very low resolution view. So again, he could not purify a complex that contained all three of these proteins, but he was also able separately to purify MER2. And MER2 is very nicely behaved by itself, but it's a tetramer, homotetrameric structure. And in the center of it, this gray region is predicted to be a alpha helical coiled coil. So this is kind of our representation of that structure. Based on the cross-linking mass spec data, he can infer that the coiled coil is probably a mix of parallel and anti-parallel alpha helices in the coiled coil. Okay, so both of these proteins bind to DNA, but they bind very differently from the SPO11 core complex, and they bind very similar to each other. So I'm going to show you data for both complexes, but in general, they show very similar patterns. So the magenta will indicate the REC114 MY4 complex usually, and the green will usually represent MER2. So here's a gel shift experiment on 80 base paired DNA duplex substrates. You can see that REC114 MY4 will shift this up kind of an indiscriminate way, indistinct way, and eventually just give you a well shift, and MER2 does the same thing. And what you can see from these quantifications is that It really likes the longer substrates better. Both of these complexes like the longer substrates better, although they will bind at least weakly to very short substrates as well. So Cornton also looked at this by atomic force microscopy, and he got a very different picture from what I showed you for SPO11. So these first AFM images are for the protein alone. So this is Rec114 MY4 without DNA or MER2 without DNA. And you just kind of see these, in the case of Rec114, pretty uniform looking particles. In the case of MER2, you kind of get this sort of flocculent looking appearance, but again, very small particles. But if you do the same experiment in the presence of plasmid DNA, you get a very different image. Instead of these little tiny particles bound to the DNA, you see these gigantic protein DNA complexes, which must have hundreds or even thousands of the proteins. What I want to draw your attention to is that the free parts of the mica surface are almost completely devoid of free protein particles. So essentially all of the protein is getting bound up in these structures, and that's one indication among some others that I'm not going to go into, that this is really highly cooperative DNA binding by these proteins. Okay, so because they make such large complexes, he was actually able to visualize these by conventional microscopy by fluorescently labeling Rec114 or fluorescently labeling MER2. And if he assembles them on DNA, he can see these small foci. If he includes a molecular crowding agent, polyethylene glycol, he gets these really big, fat, juicy foci, and the same is true for MER2. So these condensates for nucleoprotein assemblies are promoted by molecular crowding, as I show you here. What I'm not showing you, but you can look at in the paper, is that these are reversible. If you challenge them with high salt or DNase I, they'll fall apart. And these complexes confuse with one another. These little ones confuse to make bigger ones. And so these are all properties that are reminiscent of various types of phase separation systems. So that's the kind of operating model at this point, is that this is a form of biomolecular condensation. It's a DNA dependent condensation. And I do want to emphasize, I didn't really kind of point out before, but we're talking about pretty low, still pretty low concentrations, so tens of nanomolar concentrations to get pretty good binding. So this is does not require micromolar or really high concentrations of proteins to occur in vitro. Okay, so as you may be aware, there's lots of systems that show phase separation or biomolecular condensate formation. And what's really difficult is being able to tell whether that condensate formation is the functional form. Just because it forms in vitro, even just because it forms in vivo, doesn't mean that's where the activity lies, or that the activity is even affected or dependent on those condensates. It's a really difficult problem to address, and we have not proven this, but what I'll show you is some evidence that points at least in that direction, that these are functional in vivo. So what Cornton did was to try to identify DNA binding surfaces and in both of these complexes, and he was able to identify mutations in both Rec114 and in MER2 that attenuate the DNA binding activity. So this is a substitution for 4 lysines and arginines to substituting them to alanine and the C terminus of Rec114, and likewise a substitution of 4 basic amino acids to alanine in the C-terminal tail of MER2. And you can see that both of these mutants reduce the DNA binding affinity in these gel shift assays. It doesn't kill the DNA binding, it just reduces it a bit more for the MER2 substrate. But what's really interesting is it pretty dramatically attenuates the condensate formation. Again, it doesn't completely eliminate it, but it attenuates quite a bit the condensate formation. And this is what you would expect if these condensates are made up of lots of very relatively low affinity interactions. If you attenuate those interactions, you will disrupt the ability to form these cooperative structures. Okay, so of course the whole purpose in doing this was to look in vivo. So what I didn't tell you is that these proteins make very nice foci on chromosomes in vivo. So this is staining for Rec114 on a meiotic chromosome spread. You can see lots of little foci of Rec114. You can also see lots of little foci of MER2. And these DNA binding mutants pretty substantially reduce the formation of these complexes in vivo. They also kill the function of these proteins for double-strand break formation. So these are southern blots of genomic DNA that's been isolated and then cut with a restriction enzyme and probed on a southern blot. You can see that in wild type cells, if you isolate DNA at 0, 3, and 4 hours into meiosis, at 0 hours, you see only the parental length DNA fragment, but at 3 and 4 hours, you see the formation and then gradual disappearance of these broken chromosome fragments. And in a REC114 deletion mutant, there's no double-strand breaks, as we already knew, of course, and then this DNA binding mutant, even though it does not completely eliminate DNA binding and does not completely eliminate these, but only greatly reduces them, it completely kills the function in vivo. And the same is true for MER2. So these sort of point at the idea that, that these condensates might be, in fact, important for the function in vivo. All right, so I started out by telling you that these guys work sort of as a loose alliance with each other, but now I've been telling you that REC114 and MI4 make a complex and MER2 make a complex. So how do they work together? Because we know they must work together in vivo. Corrington did mixing experiments, and he was able to show that if you mix the two protein complexes together and then add DNA, and then ask whether you're able to form mixed complexes, indeed, that's what he saw. So here's MER2 condensates, here's REC114 MI4 condensates, and you can see they're exactly the same, and they correlate with each other very much for intensity. He then did an experiment where he asked if he could form REC114 MI4 complexes separately from MER2 complexes, could they fuse with one another and mix? So he mixed REC114 MI4 with DNA to form those complexes, mixed MER2 separately with DNA to form those complexes, and then mix them together. And then he would plate these mixtures on glass slides so that he could image them, but he would let them sit in solution for varying lengths of time. And if he plates them right away, the DNA will stick to the surface and immobilize it. So if you have complexes, they'll sit down on the slide and they cannot fuse with one another. But if you incubate them in the test tube first, they have a chance to bump into one another and fuse. And then when he puts them on the slide, they get immobilized and you can see them. So if he mixes these complexes together for a very short period of time before plopping them down on the slide, he sees either MER2 complexes or REC114 MI4 complexes, but he does not see mixing of the complexes. But if he mixes them together and lets them sit in the tube for a while, now he sees very nice mixing of the complexes. So these guys are able to exchange by actually fusing together, and then the contents of these complexes appear to be able to mix. Now this is really low resolution analysis of this, so I can't tell whether there are substructures in here in the mixed condensates. That's an interesting question that we'd like to explore. Okay, so the next question then is, well, okay, fine. This is REC114 MI4 and MER2, but how does that interact now with the SPO11 core complex? So very nicely, what Corrington was able to show was that these condensates, these mixed condensates, can recruit the core complex. So as I showed you before, the core complex will bind as discrete particles to plasmid DNA, and these guys will form these really gigantic mixed condensates. So we asked whether or not these condensates could recruit the protein, the SPO11 core complex protein, and sure enough, that's what he found. So he assembled the REC114 MI4 and MER2 complexes together and mixed them with the core complex, and then mixed them. And you can see that wherever he could see the RMM complexes, he was also able to see the SPO11 core complex. And he was able to show that that required the interaction between REC114 and REC102 and REC104. Okay, so I want to close with a couple speculations. Before I do that, I just want to point out that we think that this behavior of REC114 MI4 is conserved. So what you're looking at here now is a mouse chromosome pair, the X and the Y chromosomes, and this is a super resolution microscopy image. And as you may know, the X and the Y, of course, are not homologous to each other across most of their lengths. But at one end of the Y and X chromosome, there is a stretch of DNA segment that is homologous between the two chromosomes. This is called the pseudo-autosomal region. And that's the region where the X and Y have to pair with one another and recombine in order to segregate properly. And what Laurent Acquaviva, a postdoc in my lab in collaboration with Maria Jason's lab, has shown is that REC114 and also MY4 make this very prominent accumulation of the protein on these chromosomes. And I'm not going to go into detail on this, but these proteins make these really bright foci in addition to these small foci scattered across the rest of the chromosomes. And these bright foci correspond to regions where there's highly repetitive heterochromatic arrays of this particular mini satellite called MO2. So here's a fish experiment for this repeat unit of the MO2 mini satellite. You can see that there are these accumulations of MO2 mini satellites, and that's exactly where you see accumulation of REC114. MY4 is also accumulated there. So we think that these nucleoprotein condensates are actually a common feature in many species, not just in yeast, what we've been looking at. Okay, so I'll finish up by saying what we think might be going on here, which is that we think that the MY4 and REC114 and MER2 proteins co-assemble with the axis proteins to form these higher order structures, so these condensates, and that those condensates now provide a platform that can orient clusters of SPO11 activity. And again, we think that these may be sort of upside down relative to topo6, where instead of topo6 allowing DNA molecules in through the GHKL gate, we think that SPO11 complexes may allow DNA in through the SPO11 gate. So the idea is that these would then be the structures that would capture a DNA loop segment and break it in proximity to the axis. There's some interesting implications from this. One is that because these form very cooperatively, they have a tendency to basically suck up all the protein in the vicinity, right? So if you have freely diffusing REC114, MY4, MER2 proteins, they're going to be more likely to bind to an existing complex than to start forming a new complex. So the net result is that you'll actually tend to space these complexes out on the chromosomes, which might explain a known behavior in yeast that hotspots can compete with each other for break formation. We also think that they provide a signaling platform that once SPO11 makes a double-strand break, that can signal to the TEL1 kinase, which is related to the ATM kinase in human cells. And that ATM kinase, we know, turns off double-strand break formation. So we're intrigued by the idea that this structure gives a signaling platform to activate TEL1, but also to allow TEL1 to rapidly inactivate all the other SPO11s in this complex. And then finally, I'll come back to this point that we think SPO11 binds very avidly to the DNA ends, both covalently and non-covalently. We're kind of intrigued by the idea that it might remain associated with the double-strand break end while that double-strand break, the single-strand DNA on the process double-strand break, is engaging in a recombination interaction, and that this may actually help the cell coordinate the behavior of the two DNA ends and possibly also facilitate the repair. So I'll end there and acknowledge again, the majority of the work I told you was from Corentin, who has his own lab now. He had some help from Eleni Mimitu and Sam Tishfield. I want to acknowledge collaborations with Maria Jason and Dinshal Patel's lab and our core facilities, as well as with James Berger at Hopkins. And of course, none of this would have been possible without the funding. Thanks.
Fantastic. I think people may just be getting their questions in line, but we do have one question from Lu Chen Lu, which is, thank you for an amazing talk. I'll second that. Do you think that the model of the SPO11 complex being upside down could allow for a more active enzyme complex to promote double-strand break formation in meiotic cells?
Yeah, that's exactly what we think. It's kind of, so more specifically, what I think is that, you know, SPO11 is dangerous. Making breaks is not, you know, not a trivial activity. And I think the cell has probably evolved pretty elaborate mechanisms to control SPO11, so it only acts at the right time in the right place. And I think part of the way SPO11 is controlled is actually that it's evolved to be a really terrible enzyme. It just isn't very good at cleaving DNA. And so the cell can kind of overcome that, if that's correct, by putting lots of SPO11s together in one complex. So if any one of them is kind of, you know, slow rate of break formation, but you have lots of them, then you will get at least one break from that cluster of SPO11s. But what we also know is that once SPO11 makes a break, it activates TEL1 or ATM, and that then very rapidly inhibits further break formation. So what we think is that a double-strand break machine is many SPO11s, each of which is not very good at the activity, but the whole machine itself is very efficient. And once it makes a break, though, it gets shut off by this negative feedback circuit. So what you're asking about is exactly the way we think this is probably working. It doesn't bode well for trying to reassemble this in vitro, I have to admit, because it may take some pretty complicated conditions to support the activity in vitro, but you know, we'll have to see.
So I have a couple of questions. My first question is, do you think it's really necessary that the core complex be localized in a defined orientation only on the outside of the condensate, or could it be a more random thing where it's only that some of the ones on the outside are in a position to engage the substrate, and so you don't need such a deterministic arrangement of molecules in the condensate?
So at this point, we don't know. So we're kind of in the realm of speculating. I think probably it's not very deterministic. I think it's probably a bit sloppy and flexible. And I think that's part of, you know, in general, I tend to think that really highly regulated things are you know, there are things that are highly structured, highly regulated, and then there are also other things that tend to be a bit sloppier and more error tolerant. And I think that this system may be more error tolerant by evolution, because the has to be able to adapt to chaotic stochastic occurrences, meaning things like if you if the meiotic cell is heterozygous for a large deletion, say, the cell has to be able to deal with that, or you know, any chromosome abnormality is going to prevent you from doing meiosis at all. So I think the system has built into it a lot of error correction. And so I tend to think that these complexes are a bit sloppy as a way of maintaining flexibility and error correction. It's just kind of a general thought on it. But in terms of your specific question, how SPO11 is oriented in these complexes, the answer, but the reason I can show maybe one more bit of this. This calls in some a pair of papers that was just published last week in Nature. One from Matt Neal's lab, another I'm not showing you here from Franz Klein's lab, that shows that SPO11 can in fact cut the same DNA molecule multiple times. And so normally we think of SPO11 making a double-strand break and then endonucleolytically clipping to release these covalent SPO11 oligo complexes. And Matt, when he was a postdoc in my lab, discovered these covalent SPO11 oligo complexes. And these are the free end is 3', so you can label that with a terminal transferase, run this out on a gel. And so if you digest the protein after the labeling reaction, run a sequencing gel, you can see two size classes of oligos from wild type yeast cells. You see a shorter oligo class that is, I'm not showing you the standards, but these are in the vicinity of about 10 bases or so. These are in the vicinity of 20 to 30 bases. There's some longer ones as well, but we really didn't pay a lot of attention to those.
I took long
so
ones
that,
with the double cuts.
well, so yeah, so if you get rid of SAY2, which is required for MR11 nuclease activity, you should only have these covalent complexes, but you should not have any SPO11 oligos. But in fact, you get SPO11 oligos in a SAY2 mutant. We didn't know this years ago, but as we improved the methods, Matt was able to show, you actually get this very nice ladder of higher molecular weight stuff that is present in wild type. But what you're missing are these guys, and instead you just retain this ladder. And so this ladder has a 10 base pair periodicity, which of course, has interesting implications for what's going on. But what Matt has been able to show is that these SPO11 oligos that are MR11 independent are in fact from double cuts, where two SPO11s have cut the same DNA molecule, because that'll give you a SPO11 oligo here and another SPO11 oligo here that can be labeled with terminal transferase, because again, they have free 3' ends. So the idea is that the 10 base pair periodicity comes about because the SPO11 complexes all face in the same direction. And that means that a DNA molecule that then is captured by multiple SPO11 complexes, the cuts will all be coming from one face of that DNA molecule because the enzymes are oriented together. So this is part of why we're interested in this idea that the SPO11 complexes are on the surface of these RMM condensates. But that's not the same as saying that they're all on the surface. They could be also interior, and this 10 base pair periodicity could come in any way of kind of co-orienting the SPO11 complexes. So they don't necessarily have to be on the surface, although that's the idea we kind of.
Can you make big enough condensates so that you can just look directly by imaging at whether the core?
So that's in the books to do. What we have so far doesn't allow us to ask that question, but I think it would be really nice to kind of dive into that further. Yeah.
Okay, well, if there are no further questions, I just want to virtually give Scott some applause for a really exciting and interesting talk. And I'm sure he'd be happy to follow up with anybody if there are questions.
Yeah, absolutely.
Later by email. Okay, thanks everybody.
Thanks.